Well, good morning, everybody. It's wonderful to see everyone here, bright and early on a Saturday morning. I hope everyone enjoyed their light breakfast. I am instructed to tell you that coffee will be on tap all day. Do not panic if you go out and it's run out. It's going to get topped up. So I don't have very many introductory remarks to make. I, first of all, of course, want to welcome our colleague and the person who we are honoring today, Denise Park. The purpose of this meeting is to honor the contributions of Denise by having scientific presentations given by former mentorees and colleagues and collaborators of Denise. And so part of the aim today is to keep her front and center in our consciousness, but at the same time, this is also an opportunity for science and for substantial scientific contributions, presentations, and I hope for some very interesting discussions. So we've organized the program so that there will be a series of presentations, then at the end of the morning session and the end of the afternoon session, there will be two general discussion sessions which are fairly generously timetabled. And we really hope that people will step up, ask questions, argue with each other in the most polite way, of course, and we get both a lot of pleasure out of the day, and also we learn something. So that's the idea. I'm going to ask all speakers, please. You were asked to prepare a talk for about 20 minutes long, followed by a few minutes Q&A. Please try and hold to that. If you don't, our chairs, who have been selected for their ruthlessness, will undoubtedly intervene. I don't think there are any other issues, are there? If you need any guidance outside, we have ushers outside. You'll be able to spot them because they'll be the people who look authoritative standing around, and they will be happy to guide you to wherever you need to go. Let me also, at the outset here, just acknowledge right at the beginning the contributions of the CVL administrative and support team who are responsible essentially for the entire organization of this. Camilla Cristiani. You hate this, I know, so I couldn't resist. I couldn't resist the temptation. I'm sorry. Camilla and her team have worked so hard to bring this to fruition. And for those of you who know the saga of this is, I think, our third attempt to actually get something together. And we're so pleased to see everyone here, and we're so pleased that we're able to do this. So that's all I have to say. Without further ado, could I ask our first chair to come up. I'll hand over the proceedings to you. This is Dr. Chandramallika Basak, a faculty colleague in CVL, and it's over to you, Chandramallika.
Thank you, Mick. Well, it's an honor to chair the first session and have Dr. Neftali Raz from Wayne State University, soon to move to SUNY Long Island, that I heard, who's going to present research on individual differences in brain and cognitive aging, the role of vascular and metabolic risk factors. So we're starting off with a great talk, and he's someone that I've admired since I was this small, and he was this small. I'm just joking. But from my grad school days, so I can't wait to hear more about this longitudinal studies that he has been doing and continues to do. So, Neftali, over to you.
All right, so let's start this show with the show on the road. And I'm going today very briefly review the evidence of role of the vascular metabolic factors in the normal aging. So given the shortness of this talk, a lot will be left out, and we can catch with some of this in the discussion, but just to warn you that there will be evidential holes there. All right. So first of all, I would like to anchor it in the theory, in a specific theory of aging, and I think it's very important for us to keep in mind when we're talking about aging, what is that we're talking about? What is aging? Of course, nobody has a single answer. There are biologists and psychologists been working on it for already more than 100 years, and we're still not very close. But my favorite is the Tom Kirkwood theory of aging, the so-called disposable soma, which basically states that aging is not, unlike development, aging is not a programmed process. It's not unfolding according to some plan. There is no crawl first and stand next and walk later type of progression. And what basically, according to this theory, what basically happens is that it's a stochastic process of wear and tear. So as the time passes, especially in the post-reproductive aging, there is no evidence that there is any evolutionary pressure of getting old. There's no advantage for that. So we basically course, and we course to the end of our lives, subjected to all sorts of random influences, either physiological or environmental. And gradually the soma, the substance, is wearing out. So that's the main idea. So how this wearing out is happening, again, it's a very influential theory proposed by Harman very long time ago, so almost 65 years ago, that basically said that the foundation of cellular senescence is basically the activity of the so-called free radical or reactive oxygen species. And these reactive oxygen species, they're always present. It's not some sort of aliens landing in our cells and doing damage. They're always there. They do a lot of important and useful things. Only with the passage of time, this equilibrium shifts, and the mopping up of the reactive species also is not catching up with the production of those oxygen species. And it's not only oxygen, but there is actually reactive nitrogen and carbon species that are playing the role. But it's possible, of course, that it's not that the aging is programmed, but it's possible that the old programs just keep running. That's possible, but it doesn't make aging a program process. So again, it's some sort of a random wear and tear that takes its toll. Now, why do I think it's important? I think it's important because we're dealing with the brain, and the brain, which is really insignificantly small part of the body, draws the very significant share of resources, mainly energy, blood flow going to the brain, glucose consumption, etc. And all this, of course, is necessary mostly for neural transmission and repair of the structure of the brain. So what we're talking about is eventually gradual wear and tear of the informational hardware of the brain. And just to remind you that the brain is basically a jello that's sitting in this tight basket of blood vessels. And therefore, anything that affects those blood vessels, anything that affects their structure, their function, their ability to deliver the products and remove the waste will affect the function and eventually the aging of the brain. So we know that vascular risk increases with age. And if you are in the late 70s, your chances to escape hypertension are very small, though possible. And we know that hypertension in the middle age, not in the older adults, but in the middle age, is a predictor of later decline. There are multiple studies on that. I'm just quoting one of them, but there are others. Metabolic risk in middle age, even when we're not talking about diabetes, we're talking about metabolic risk as a conglomeration of factors that at the extreme appear as a metabolic syndrome. But not even a full metabolic syndrome. Metabolic risk in middle age predicts lower cognitive performance in older adults. And even a normal but high glucose is already a risk factor for cognitive decline. Again, this is a meta-analysis of multiple studies that point in this direction. Those effects are not very strong, as you know, but they're consistent. And in tune with the disposable soma theory, we can predict that those multiple small influences add up. Even genetic factors that are associated with cognitive decline, like ApoE, for example, those are vascular genes. And ApoE, very significantly, the load of the quote-unquote bad ApoE epsilon 4 allele is tightly associated with the increase in cholesterol. Cognitively, also, it had been noted a long time ago, and this is a sort of a stark statement that by neurologists like those very tough, stark statements, and this by J. Alpert, who said that he noticed that the impairment of cerebral functions equivalent to the patients with a surgical removal of both frontal lobes can be seen in the people with hypertension. Okay, he exercised the truthful hyperbole, but nonetheless, that's a direction that the significant increase in blood pressure, and especially pulse pressure, is associated with impaired cognitive function. All right. So I find it very useful to see it as a conglomeration of factors, as Wadley called the vascular health triad. So it's not just one factor, one indicator. And I know you can open any medical journal, there will be something about a single indicator that popped out significant. That doesn't seem like a good, sort of philosophical, it doesn't seem like a good way of dealing with the problem, because we cannot smoothly glide from one indicator to the construct. And whenever we're talking about vascular risk or metabolic risk, we're talking about the construct. And so those things need to be converging. And even more so, the vascular, metabolic, and inflammatory risk, they all seem to converge in drivers of aging eventually. And the problem, of course, is that they all affect the vascular system, including cerebrovascular system, and they produce endothelial dysfunction. The vessels become less pliable, less responsible, and as the atherosclerotic process continues in the blood vessels, it creates sort of a vicious cycle. There's more inflammation, more atherosclerosis, less blood flow, less responsivity, and all this happens while increasing the reactive oxygen, nitrogen, and carbon species burden. It's a vicious cycle. So on this high note, we can continue to look at the findings, which is always tougher than theories. But I just put together several findings from our lab, and there are, of course, multiple people working in this area, so it's not our exclusive scoop. But we found, for example, that people with hypertension show significantly greater shrinkage, regional shrinkage of the brain, that iron accumulation, iron, which is a marker, actually a significant marker of reactive oxygen species activity, is associated with hypertension, that homocysteine, another vascular risk factor, combined with aging, accounts for an increase in the white matter hyperintensities in the brain, that low cerebral blood flow and outflow, we measured in that particular study the flow to the brain and flow out of the brain through jugular veins. They all related to the vascular risk factors. Again, those are not strong effects, but they're there. And with inflammation, it's actually tough to measure, to relate the blood marker of inflammation to what's happening in the brain. But if you look at the risk factors, the genetic risk factors for inflammation, you see that they associated with the faster shrinkage of the brain regions. And the increase in iron seen longitudinally is coupled with the shrinkage in the basal ganglia. So there is plenty of evidence that even in healthy, and this is all in healthy adults, screened for just about anything except hypertension. So if you look at the population studies, those effects are much larger. But then you have to, of course, control for multiple other factors. This is another study. This is by Anna Doherty from her dissertation, when look specifically at the metabolic risk, which in this case it's a factor, so it's a real construct from 5 different indicators which converge very nicely. And you can look here and see that the metabolic risk, increased metabolic risk, of course, is associated with age, but also is associated with the increased accumulation of iron in the putamen, which in turn drives the longitudinal increase in iron, and which is coupled with the reduction in the putamen volume. So in this study, we show that there's actually a chain of events that happens in a relatively short time, in this case, 2.5 years on average. One time. More recently, we analyzed some data when we looked at the hippocampal volume and the blood flow in major cerebral arteries by using ultrasound. This is the transcranial Doppler ultrasound measure, where you can measure the blood flow, but you also, for us, the important point was that you can measure the pulsatility index, which is a good proxy for endothelial dysfunction. And it's a small sample, but what you see here, there is this nice correlation between the shrinkage of the hippocampus and increase in pulsatility. And you can see people who didn't show increase in pulsatility, there is really no relationship. But then you see this clear relation, those folks who actually did. So, I tried some time ago to put it all together in one big thing, one big boxology model, because obviously everyone needs to have some boxology model, otherwise you're not really taken seriously. Of course, I may be not taken seriously even after that, but at least I tried. So, okay, so where is the initial event? When does it all start? And it probably starts with a random mutation on the very cellular level in the mitochondria, the energy factories that produce, that are responsible for production of ATP. And that, of course, mitochondria are present in all cells, and they're responsible with aging for sarcopenia, for reduction in muscle mass, and also probably for the brain decline. And mitochondrial dysfunction, of course, is associated with an energy deficit because it produces the accumulation of reactive oxygen species. In turn, they induce greater deterioration of mitochondrial genome and mitochondrial dysfunction. And this energy deficit causes accumulation of iron, which we have demonstrated, and the iron accumulation in turn creates all the, continues the induced accumulation of reactive oxygen species. Now, the vascular part comes from, of course, endothelial dysfunction that includes reduced blood flow, reduced glucose metabolism, and eventually reduced synaptogenesis through inflammation and reduction of blood flow. And this specific part here that I added, the environmental pollution stress, that's a more recent work that shows that a lot of those factors are actually associated with increase in environmental particulate pollution. There's very little human work yet, but in rodents, there's a clear evidence that being placed in a polluted environment causes reduction in the hippocampal volume. And then, of course, myelin maintenance, that you need a lot of energy to do it, and you need to maintain myelin through the whole life. Membranes need maintenance also. If you experience myelin loss and neuropil loss, which neuropil loss and probably to some extent intracortical myelin loss account for all those volumetric findings that we replicated many times. Structural connectivity loss is, of course, associated with both reduction of the integrity of the neurons and myelin repair. That reduces the functional dynamic range, meaning that I didn't have time to put this slide here, but we found recently that if you look longitudinally, people who show reduced variability of resting fMRI signal are the people who show either cognitive decline or inability to benefit from repeated cognitive testing. And eventually we come to the cognitive decline that we also like to study. Now, in this busy scheme of things, what do we know? We do know, of course, of cognitive decline. We know that there's a reduction in dynamic range of the brain. We know there's a drop in signal-to-noise ratio. So this part we're already tied together nicely. We know on this end that all the vascular things are associated also. We know that mitochondrial dysfunction happens with age. What we really need to know, we need to demonstrate that energy deficit ties it all together. And it's, of course, easier said than done. There are ways of assessing brain energy metabolism, but they're not yet as clear and differentiated as we would like. So we now collecting some data on using phosphorus spectroscopy, trying to get into energy metabolism and into turnover of membranes with age. But it's on 3T, and I wish, of course, I could put my hands on 7T at some point and do more thorough examination of this. All right, I think I'm right on time, surprisingly. And just to mention that we do want to know more about the effect of metabolic and vascular risk on aging process, because they can be managed. They can be alleviated, mitigated through multiple exercises, including entertainment and even yoga. And of course, don't forget to keep your stress low. Many thanks to all the people who did all the work while I entertained wining and dining in conferences, and to kiss the hand that hands me the money, of course. Thank you very much.
Thank you very much, Neftali. We have time for a couple of questions. Yes.
I have a question. At the box model, you put a time down there.
Can you speak louder, please?
In the box model,
Yes.
that you showed that you have like these different physiological changes as a function of age. What is the time scale? What do you envision as the time scale for all these processes?
The reason I put the time axis there is not because I know the time scale, but just to show the temporal order of things, supposed. Time scale, it's anybody's guess. I mean, it can start pretty early. We know there's evidence, at least from the vascular factors in the middle age, affecting the cognition 20, 25 years later. So it happens as we speak.
Yes.
Hi, great talk. A couple things. So, what you reported to in terms of energy deficit and such, you know, those things have been are quite hot topics in just the biology of aging these days. And of course, there are other hallmarks of biology of aging. And what I liked about yours is you're actually included more than one, you had 2.5 maybe. But one of the ones that was interesting and sometimes I think could lead this to become an interface to abnormal cognitive aging is in fact, you know, proteostasis. So do you have any thoughts about how you would incorporate, you know, abnormal proteostasis, whether it's, you know, accumulation of, you know, aggregates or anything else and this kind of thing?
Well, I'm not a neurobiologist,
No.
I'm not a molecular biologist, so my understanding is that any processes that require for folding and mopping of proteins, they're energy driven. So it's all maybe traceable eventually to the energetic dysfunction.
Ooh, that's good, that's testable too. Yeah, there you go. Write that one.
Any further questions? I have one question. As I was doing the research on pulse pressure and its relationship to fitness, there have been confounding studies. Usually people think that, oh, if you are high fit, you should have low pulse pressure, or even interventions. But there have been studies shown in young that if you actually do strength training, then it actually makes your arteries, more stiff, and you have increased arterial stiffness. So, it's not like that
Yeah.
you do one, and on the other hand,
Yeah.
Tai Chi training seems to,
Yeah.
make it more flexible. So, any thoughts that you have about
Well,
this?
I mean, it's a usual situation with too much of a good thing is never very good. For example, with aging, aerobic training is fantastic, and aerobic exercise is very beneficial, except that at some point, orthopedics catching up with aerobics, and cartilage doesn't last forever. So that's why, I mean, it's very important to balance different types of activities. So some strength training is very important. Like if you don't have a decent core, you're not going to run well, you're going to ruin your limbs, sort of, to put it very bluntly. But overdoing things never works. And yes, in weightlifters, in like competitive weightlifters, it was shown that there is an increase in arterial stiffness. It's more complicated because also there are some studies showing that insertion deletion of the act actually with the people with double deletion are more prone to that sort of effects.
Well, thank you very much. And thanks again. So our next speaker is Dr. Gerard Bischoff. He's going to talk about multimodal imaging in the borderland of aging and beyond. And just a little tidbit, I really miss Gerard and CBL because we won so many mixed doubles badminton championship together. And as you see, after Gerard has left, I have inflated because I've not played much badminton. But yeah, it's lovely to see a friend back again.
Okay, well, good morning, everyone. And Denise, thank you so very much for this invitation. And it's really great to see all of you here again and being in this in-person meeting. And yeah, I'm working on multimodal imaging in the borderland of aging and beyond. And Denise and I actually met in 2006, I believe, at the University of Illinois. And we've been friends and colleagues ever since. I then moved with her to the University of Dallas, and that's when we started the Center for Vital Longevity. And I'm really delighted that I'm honored to speak on what we have accomplished. And so one of the goals that I'm looking at is multimodal imaging and using MRI PET imaging to understand what does cause neurodegenerative disease. And when I'm thinking about Denise's and my collaboration, it's really this sentences taken out of the Cognitive Neuroscience of Aging book that she co-authored and co-edited, is the combination of different MRI measures or MRI and PET markers is today quite common. It's a powerful approach where the sum can be greater than the parts. And it was really that statement and sort of that sentiment that Denise represents, as well as with Kristen and Karen, that had drawn me to her laboratory, and that was one of the reasons why I decided to work with her. In the preparation of this, I was thinking, so how do you actually give a Festschrift presentation, and what do you say? And so I've been thinking, I was thinking back in 2015 when Angela Gutches and Eric Leschiker reached out and asked us to provide a letter for Denise's mentorship award that she was then awarded by the Division 2020. And I don't think that you ever read the letter, and so I actually put some of the sentences in this presentation. So for me, one of the very unique talents and traits of Denise is her foresight, specifically when it comes to the creativity of her scientific ideas and the innovative nature of her program of research. So one of the things that we learned from autopsy study is that there's an age-related increase of beta-amyloid pathology, and beta-amyloid is the hallmark of the Alzheimer's disease pathophysiology. And ever since this opportunity to be able to image it using positron emission tomography, we were quite fortunate now to actually look at these age-related associations. So some of the first initial studies that looked at healthy older adults found that there is a proportion of individuals who have some type of amyloid pathology in their brain, very much similar in the amount to the Alzheimer's disease patients, but they are totally far away from any clinical symptoms and far away from the clinical onset of Alzheimer's disease. So this is a study that I really like because they followed up these individuals who were amyloid positive at baseline or inclusion of the studies over 8 years. So one of the longest longitudinal study actually done on this topic. They also included individuals who showed significant neurodegeneration in terms of hippocampal atrophy. They also included individuals who showed both, so positive on amyloid and neurodegeneration or hippocampal atrophy, as well as individuals who only had hippocampal atrophy but no amyloid. When they looked at the progression of these individuals over time to progress to either MCI or Alzheimer's disease, they found that it were the individuals who were positive on both biomarkers at inclusion of the study, as well as those individuals who were amyloid positive at baseline. So this indicated to us that increased beta-amyloid burden alone confers a greater risk for the clinical diagnosis of Alzheimer's disease than hippocampal neurodegeneration alone. So one of the problems with all of these studies up to then our work was that the majority of these individuals were quite old. 73.1 was the mean age of inclusion of the study here. And that's what the foresight was that I feel like Denise, Kristen, and Karen Mike DeVoe had put together to try to include beta-amyloid imaging into the Dallas Lifespan brain study and start imaging these people much earlier, actually at the age of 30. And Karen was showing that there was already quite a significant age-related increase of beta-amyloid pathology measured across the human lifespan starting at the age of 30. And what I did then in my dissertation work, looking specifically at these individuals who were 30 to 49, to see if some of this age-related increase was associated with memory performance or cognitive performance, and indeed it was associated with both recall and recognition performance already at the ages of 30 to 49 in these individuals. So at the time, a lot of people thought, okay, I don't believe it. It's maybe selection bias or whatever. It's the Dallas Lifespan brain studies. There are a lot of highly educated individuals. So I was really happy to see that this was actually replicated in a French cohort by Gail Shedler's lab, who included even younger individuals, starting from the ages 20 to 60, and found the very similar finding, an age-related increase already at this early age. But she also found that the regions most vulnerable to either the age effect compared to the regions most vulnerable to the Alzheimer's disease effect were different. So the Alzheimer's disease pathology was found to be more frontal versus the age-related effect was more located in temporal regions. So there seems to be an age-related increase of beta-amyloid burden that's regionally distinct, depending on which perspective you're looking at, from the AD or from the normal aging perspective. Then there was a lot of work that Denise did with Michelle Farrow, Karen, and Kristen, and looking at following these individuals up in a longitudinal fashion, and then showed that there was indeed a continuous effect of beta-amyloid on memory, longitudinal as well. And there was Heidi Jacobs and me at HI in 2018 who thought about what does it all mean? Like, is it really, like, how can we justify that there's a threshold that sort of indicates Alzheimer's disease pathology if we have this continuous evidence that if you look at it continuously, you still see an effect on this variable. So we are thinking about what could promote beta-amyloid pathology early on, and it's Karen's work who looked at hypertension, who showed that, this could be actually a modifier of beta-amyloid pathology in normal aging. And we were sort of advocating for this idea that, like, what determines the increase of beta-amyloid pathology and across the lifespan that we put down in this proposal, and what are some of the risk-promoting or risk-reducing factors that may relate to this continuous buildup of this Alzheimer's disease pathology. So when I think about all of this and put it together, I feel like the seed was sort of, started here in Dallas, where we found that the accumulation of beta-amyloid pathology is age-related, sensitive to age in the lifespan sample of DLBS, and that it increases with age with consequences to cognition, that in middle-aged adults we find that beta-amyloid burden has negative consequences to memory, and that sort of culminated into this new research proposal on the biological and clinical meaning of subthreshold beta-amyloid in normal cognitive aging. So one of the things that Denise has taught me is really how important it is to work as an independent researcher early on, and how to juggle multiple responsibilities, which helped me adjust my work style more efficiently, and prepared me for the responsibility as an independent researcher in the future. And that's really what it is. We were tasked with a lot of responsibilities, and I'm seeing this now in my current job, that it really helped me to adjust to this. So then I moved to the University Hospital Cologne, Germany. And what sort of was interesting to me doing this switch was looking at the disease rather than the normal aging end of things. And So you all have seen these curves with this age-related, with this disease-specific increase of beta-amyloid pathology that precedes changes in tau that we see that then follows into pattern of neurodegeneration. So the first thing that we did, well, I did in Cologne, is they did this multimodal imaging studies looking at triple PET. So we imaged individuals with Alzheimer's disease patients who were, and with amyloid imaging using PIP, with tau imaging using AV1451, and looking at the glucose metabolism pattern using FDG PET. And first we found this overlap, or this sort of ubiquitous distribution of beta amyloid across many brain regions in Alzheimer's disease patients, but a very regionally specific pattern of this tau ligand that we used in the Alzheimer's disease patients. It's very much aligned with the reduction in glucose metabolism that we were seeing in these patients. And if we look at this across the regions of the brain, there was a positive correlation, and that was actually quite high. And but there was also an interaction with amyloid. So both sort of protein pathologies seem to contribute to neurodegeneration measures using FDG PET, in the sense that regions who had less amyloid, the relationship between FDG PET and tau wasn't as strong as compared to regions who had who had more amyloid, where we found a much stronger relationship between FDG PET and tau. So when we looked at this data and we sort of started to sort of group these individuals by their clinical severity, we found that eventually at the beginning of the disease, there's one regional specific activity of this ligand in the brain of these patients, but it sort of spreads across many brain regions once the disease advances. And so one of the questions was then, what determines the spread of tau pathology in Alzheimer's disease? And that was really the work of my colleague and our PhD student at the time, Merle Hönig, who looked at our tau PET data of these patients as well as resting data from RAI. And what she did, she used an independent component analysis on the tau PET data, and she identified 10 different components in the tau networks that we call them. And then we looked at the peak voxels, the peak regions of the peak regions in these networks, and we used that location as a seed in resting state connectivity data to sort of identify which are the regions that this peak is connected to. And then we looked at what's the overlap between the tau pathology and the region that this peak region was connected to, and the overlap looking at a dice coefficient was quite high. But it also overlapped with known resting state networks such as the ventral default mode network. And we interpret this as sort of at least initial evidence that tau deposition occurs independently in a different set of regions that are functionally connected to each other. And that sort of motivate this idea that maybe the spreading of tau throughout the brain is somehow facilitated by the functional connectivity between these regions. So we moved on we continued this line of work by looking at the relationship between resting state connectivity measures and tau pathology in Alzheimer's disease. This work of my PhD student, Anna Weller, at the math department in Cologne. And what she did, she looked at the community structure of resting state functional connectivity data in controls, young controls, older adults, and Alzheimer's disease patients. And so there are different things that we noticed. First, that the number of communities was different in Alzheimer's disease patients compared to senior controls and young adults. And then we looked at different aspects of the community structure, and we turned to Dallas, actually, to this fancy group of people who identified the segregation as a meaningful measure of describing community structure or describing networks. And so we found that the segregation coefficient was significantly lower in the Alzheimer's disease patients compared to the other groups. We also noted that there were nodes that had left their original community, if you use the young controls as a reference, and then there were nodes that remained in the network compared to the young controls. So we basically looked at the nodes that had left their lesions as a function of disease compared to those who stayed, and we looked at the tau in these nodes. So we had tau data on these as well. And so we found that the nodes that left sort of the community over the course of the disease also had more tau. There were more tau pathology in these regions, or SUVI in these regions. So to summarize this part, the multimodal imaging in Alzheimer's disease that I did then in Cologne revealed a close relationship between in vivo measures of tau pathology and in vivo measures of glucose metabolism. We also found these interactive effects of beta-amyloid on tau and regional glucose metabolism. And we found this overlap of tau in regions that are functionally connected to each other, indicating a potential spreading mechanism. That's still something that we further explore. And that the changes in the community structure of functional connectivity data in Alzheimer's disease somehow, at least to a certain extent, does relate to increased tau pathology in this region, specifically those regions that switch their allegiance as a course of the disease. So one of the things that we heard yesterday in the symposium about Denise is that, I can only attest to this as well, is that Denise, and I think we all can, is that Denise has been an incredible, generous advisor who offered me a host of opportunities in these 5 years, including international conference travels, graduate level teaching, workshops, networking opportunities with both national and international scientists that really greatly shaped me, not only my academic thinking, but also me as a person. So I continue this work on protein pathology and neurodegeneration, and one of the reasons why I do this is that if you look at the different phenotypes of neurodegenerative diseases, you notice that the majority of them are somehow related to specific proteins that seem to accumulate or that seem to misfold as a function of the disease. And I focus on tau because tau is not only sensitive to the Alzheimer's disease pathology, Alzheimer's disease clinical phenotype, but it is also sensitive to other disease phenotypes such as atypical Parkinson's disease, PSP, or even the frontal dementia spectrum is associated as a primary, or is defined as a primary tauopathy. So one of the work that we then did is we looked at how can we use tau imaging as a means of a differential to identify or to make a differential diagnosis based on the protein phenotype. And so what we did is that we included different types of neurodegenerative diseases, like typical Alzheimer's disease, leukopenic variant of primary progressive aphasia, behavioral variants of Alzheimer's disease, behavioral variants of frontal dementia, and these are all tauopathies. But what's different about these diseases is that some are amyloid positive and some are amyloid negative. And so we wanted to know, can we look at tau images and identify exactly these groups? Can we dissociate these amyloid positives from the amyloid negatives? And to do this, we used the scaled sub-profile modeling approach in combination with the principal component analyses. And we identified patterns that are very sensitive for the amyloid positive group and the amyloid negative group. And then we did some we used a classifier to use these patterns to classify these groups of patients into amyloid positives and amyloid negatives. And the classification accuracy was quite high, and there are some limitations about the studies because we did not have a cross-validation sample that we really wanted to use as well. But it seems that there's something in the pattern of tau that is somehow sensitive to identifying individuals who were amyloid positive and who were amyloid negative. And to a certain extent, it matters for the disease course, for the type of interaction that you have with the patient, if it's a behavioral variant of frontal dementia or if it's Alzheimer's disease. So it seems that the pattern of tau correctly identifies amyloid positives from amyloid negative cases, and we really are following up with this now in a cross-validation cohort. So for me, imaging protein pathology can reveal the potential driver of the cognitive impairment in the clinical phenotype. I think it can guide differential diagnosis, and it can assist in the design of targeted interventions for disease-modifying therapies. And for me, what I took away from Dallas is that I continue this work on trying to integrate different markers of protein pathology, molecular imaging, in addition to looking at different markers of structural functional connectivity, as well as metabolic demands using FDG PET or perfusion images to understand what promotes protein pathology spreading in disease, Alzheimer's disease specifically, and how does it explain different clinical phenotypes. And that's really what we are trying to do, like trying to understand using these in the cross-sectional study currently that is also a longitudinal study to follow these individuals and to start quite early in the lifespan to see if we can use multimodal imaging to understand these relationships. And with that, I have to thank a lot of people. This is my lab back in Cologne and the people that I get to work with every day. And I also want to thank some people here in this room that have touched my life throughout my time here, and it was difficult to squeeze them all into this one picture or one image, but I tried. So it's definitely Kristen and Karen, Sarah Yu, who is at Chicago, then Josh Go, I visited in Taiwan. It's this group that I really enjoyed working with and collaborated with, and of course my dear friends Jenny and Derek, who are here today. And foremost, I want to thank you, Denise, and thank you all for your attention.
We have time for a couple of questions. Thank you, Gerard.
Hey, Gerard.
Hi.
Fabulous talk.
Thank you.
Wonderful work. I'm going to start with a nitpicky one. Going to your FDG and tau relationship, was that partial volume corrected?
No, it was not. These data were not partial volume corrected, but we did some partial volume correction in the follow-up studies of these patients, or like with a bigger sample that we had, and we did not see any significant change of this relationship anyways.
Okay.
Yeah.
So just to know, it's very important to be aware, since FDG is
Right.
thought to be a marker of neurodegeneration, the other major marker is cortical
Right.
thinning.
Right. Or atrophy,
Yeah,
difference,
atrophy. But so that could be a primary driver of that relationship. But more...
You think, wait, so you think that the primary relationship, meaning the relationship of tau and volume, or like you think that the...
Well, if tau kills cells,
Right.
it drives them down, the cells can't metabolize anymore, so they are
Right.
lower on FDG, right?
Right, so well then you may be aware of the studies who looked at tau and MRI and atrophy as a function, and looked at this relationship, and they found that the relationship is similar but not as strong. And so I think that not all of this is infected by atrophy, but because I feel like that at least the loss of the function of glucose metabolism of the cells seems to occur earlier before the cell dies, right?
Okay. Yeah, I was just using the extreme. But so more broadly, one of the things that I'll bring up in my talk is how much of these markers of neurodegeneration of the Alzheimer's type, so amyloid, tauopathy, FDG loss, are explaining the cognitive changes that we have previously attributed to aging, and how much can we really distinguish aging in general as having a cognitive consequence from preclinical Alzheimer's disease? And what do your data say about that?
Okay, so I think in the face of preclinical Alzheimer's disease, if you don't see any significant differences between the preclinical AD population and your normal aging population, I do believe that biomarkers can tell us something about their potential cause of their cognitive trajectory down the road. And that's what the data show with, from Victor Villemania and the Australian biomarker study, that these are were the individuals who were positive on amyloid with the second highest risk to progress to MCI and Alzheimer's disease over the course of 8 years. So the question is, can these biomarkers distinguish these two ideas, right? Preclinical and normal aging? And I think to a certain extent they can. But you're right, there is normal aging is there is beta-amyloid pathology in normal aging, and and not all of this has to go down south. They may actually never develop Alzheimer's disease. Therefore, the sensitivity of these biomarkers is definitely not as great if you look at the whole lifespan, because we still have these individuals whose head is full of amyloid, and they never get Alzheimer's disease, right? So
definitionally, they have Alzheimer's
Right. By definition, by the biomarker definition, they have Alzheimer's disease, but they don't show the clinical symptoms. Right?
It's like sufficient but not necessary conditions, or necessary but not sufficient.
Karen?
Just a tiny footnote on Sherry's comment and question is that thinking about the relationship between these biomarkers and neurodegeneration is really muddled by a very messy literature that defines neurodegeneration by a single snapshot time point of hippocampus volume, which is
Right.
absolutely an inappropriate inference to make. I mean, there's some literature that does maybe show change, but a large portion of the literature focused on these kinds of questions is looking at a cross-sectional snapshot, right? And also putting it in a box. And so I think maybe we don't have sufficient data to really address this in the way we want to in terms of delineating groups, normal and pathological.
Right, and I think that's sort of this the debate of using this cutoff. Like, do you capture all individuals who may be on the path for pathological aging if you identify a SUVR of if they have an SUVR more higher than 1.1, and like your work, our work, and the work on the continuous measures showed, no, actually not. You're still seeing cognitive decline in individuals who are sub-threshold or below that threshold. So one of the things is, and then another issue is this comorbidity. Like there's, Alzheimer's disease patients rarely just have amyloid and tau in their brain. They have Lewy body disease, like Lewy body is in the brain, TDP43. And the question is now, what does this mixed pathology do to this aging process? Is it that at the end, these individuals who have multiple pathologies in their brain are the ones who are declining faster? Maybe, but there's some evidence that actually shows that, there's some evidence that actually shows if you have an Alzheimer's disease brain, but you also have dementia, you have Lewy bodies, you're actually not declining as fast as you would if you just have pure Alzheimer's disease pathology in your brain. And so we still really don't know yet how to interpret these findings, but it comes up over and over again that mixed pathology not necessarily is a bad thing for everyone. Right?
We have time for one more question. We still have 5 minutes. Yeah.
First off, that was a great talk.
Thank you.
And I love the way you organize it, and it really kind of highlighted the influence that CBL has had on you. And I first want to say we're really proud of you.
Thank you so much.
So obviously my question is going to be about FC. So the patterns you're showing were tau ID constrained or related to some FC patterns. As you know, the rest and state correlations are not reflective of monosynaptic connections
Right.
in entirety. I just want to hear your thoughts about what you think might be causing, or whether you think there is some causal relationship between correlations at rest and spread of tau, or what you think the mechanism might be that would allow us to see these relations.
I mean, that's a great question. So I think, residing to connectivity, functional connectivity, is somehow, I guess, easy to just say, okay, if you know that something is spreading, which multimodal imaging modality are you using? And functional connectivity was the one that we felt would be closest to the question to answer it. But it's noisy. I mean, we know that it's noisy, and we know that we're going to have to replicate this in a different cohort. And I mean, there are times in my life that I did not trust functional connectivity at all. And so, and then we put this study together. I mean, the ICA study, the first one that we showed with the seeding, I thought that was a nice way to sort of get at the independence of tau and look at the seeding. And what we did, I mean, we seeded the networks in healthy controls, and we didn't seed it on the same data set, right? And then there was always this question, well, but what if it looks like that tau seems to be more likely in these regions that's connected to this seed, but then Anna showed that the regions, that the relationships or that the networks look totally different now in Alzheimer's disease. So what was first? Like, was it like that the tau pathology changes the structure of the networks? And that's what we see with Anna's data, but then it's still healthy enough, so we see the spreading pattern. So like bringing this together is really complex because, and that's what we see too, like that there's this reorganization on the one hand, but then we still can somehow show that that this spreading seems to be a pattern in healthy control. So what we're now doing, actually, we do collect longitudinal data. And we try to really see, okay, what's really the change of individuals who have MCI eventually progress to Alzheimer's disease, and really map the networks across the disease, and then see how does this relate to tau.
Well, thank you very much. We'll wrap up this session now. It's 3 minutes before the time, so it's 10:02, and we'll come back in, I think, 15 minutes or so, 10:20. Let me look at the calendar. Yeah, at 10:20 a.m., so we have ample of time to chat and discuss. Thank you very much, Gerard and Neftali.